So there's the actual data. So dormancy is something we all aspire to, okay, when it comes to cancer. And it's been something that it's been hard to define, and it has been awfully hard to, we don't know what induces dormancy, we don't know what breaks dormancy, it's just been a black box. The best investigator that I know of who has studied metastatic dormancy is Ann Chambers in Ontario. Anybody know her? Couple, yeah. She had the cutest idea. She took tumor cells, and rather than put in some genetic marker that makes them color, she put in these little beads that glowed. And that way, if the tumor wasn't dormant, if the tumor cell was going to split into 2 tumor cells, it would apportion the little beads to each of the daughter cells. And 2 into 4, you'd dilute out the beads, so that the only thing that would show up in an animal as glowing were the dormant cells that hadn't divided and start diluting out their little beads. Does that make sense? And that's what she found. She could find these beads, this is in the liver, of these tumor cells. She could take that liver out. Tumor cells will grow in a culture dish. Those tumor cells will come out of the liver, grow in a culture dish. She could re-inject them in a mouse. They'd form a primary tumor. They'd go to the liver and stop growing again. We have no clue what that's all about. Do you mind if I take this off? But, yeah, who said that? But we need to figure this out, okay? So here's the experiment she did. She took 2 cell lines, one of which was a very aggressive metastatic cell line. The other is a cell line that had these properties of dormancy. Injected them into mice, and then treated with adriamycin or doxorubicin, okay? You know the drug. Here is the effect of this drug on metastases from that aggressive cell line, okay? It makes sense. Doxorubicin decreased the metastatic burden because it is a cytotoxic. If those cells are aggressive and they're trying to grow and they're proliferating, it nails them, okay? Here is the unfortunate reality of dormancy. If the cells were in dormancy and they weren't cycling, a cytotoxic isn't going to touch them. You know, if the idea is to prevent proliferation, that's what most of those drugs are, and these cells aren't proliferating, game's over. It had absolutely no effect. And what that says is we can go through our chemotherapy, but to the extent these cells are sitting there dormant, they may be just avoiding the whole 9 yards. We wanted, as much as I would love to do one animal experiment and say, hey, we got something, we had to look in a second model system. And we pulled out another triple negative cell line. Here is the tumor growth in the mammary fat pad vehicle versus this LPA1 receptor inhibitor. No difference. Then we put them in the tail vein to get lung metastases, and it repeated. Okay, here's the data, and you can see some of the lung metastases. They are the bright yellow lesions, and here would be in the treated. So it worked in 2 models. In fact, if we took the mice and asked, how did they just survive? The vehicle-treated mice, this is the Kaplan-Meier, so it's the percentage alive over time, started dying, as you can see in the orange. And we have lots of regulations. This was breathing difficulty. The mice who got the 0719, at least till this experimental endpoint, were mostly alive. We had a third arm that got 0719 to this point right here. And then we took them off and put them on vehicle. And you can see that they then, okay? And it was pulmonary. Vehicle's placebo. It's whatever you diluted your drug in. Sugar pill. Okay, and here it is actually when I showed you dormancy. This is this second model. So here's Ki67 positive lung met, vehicle treated. the Ki67 negative lung on the 0719, this would be a lung met right here, another one right there. I don't know where the rest might be. And here are the ones where we switched them over to vehicle. And you can see larger mets, and they've returned to cycling. Why isn't this drug in the clinic? I'm sure is what you're all saying. You know, you're going to hand this out at lunch or what, Pat? Okay, so here's the inhibitor we used. It has one problem, there's not a drug without problems. It's not orally available. And I just showed you, you're going to have to take this stuff. And so the company, in realizing that it had the right biology, but they couldn't make this stuff accessible, gave up the license on it. The original compound was made by the Kirin pharmaceutical company in Japan, who makes Kirin beer, for those of you who have eaten sushi recently. And so it went back to Kirin. Never mind, undaunted, a paper came out about a year ago from a company that I'd never heard of, these little biotech companies called Amira Pharmaceuticals. And I almost passed out when I saw this paper, and they reported a variation of this drug, Amira 0095, that was orally available. And it was being developed not for cancer, but for a disease called lung fibrosis, idiopathic pulmonary fibrosis. Anybody heard of it? So this is a lung disease where the fibroblasts in your lung proliferate and proliferate and proliferate, and it just destroys lung function. Very similar to liver fibrosis, things you've heard of. I was on a plane as soon as NCI ethics let me. I paid myself to go there because they were having a fit. We negotiated with them. Thing I forgot to tell you. If you do a metastasis assay in mice with fibrosis, they get more mets. It's the same thing. Okay? We negotiated with them for about 4 or 5 months. We were down to the lawyers. We were down to a couple words here and there. Believe me, I have ethical guidelines so I don't end up in front of Congress, so it's a tough thing. And as we were down to a couple of words, the entire company was bought lock, stock, and barrel by Bristol Meyers Squibb, start over. And what they told me, you're going to like this. No, you're not. Is, Pat, we are not developing this drug for 2 indications. Okay? Because if you develop a drug for fibrosis and for metastasis, if something goes wrong here, the mice die, you still have to tell FDA for here. Okay? And they're playing it safe. They're not telling FDA a thing they don't want to tell them. Okay? So that drug died. I was then complaining to Ann Chambers, who had one of her friends. This is what your friends do for you. Your friends run patent searches. I didn't even know how to do it. And found an unpatented inhibitor, patent unpublished inhibitor at Sanofi Aventis. It is orally available. It has been through phase 1 for fibrosis as well. And all I can say is we're negotiating, so pray for me. Okay?